we'd like to get the final session underway. So our first presenter is Ágnes Peragovics from Eötvös University in Budapest. She did her master's at the Technical University of Budapest in 2009. That's when she finished, and then she started doing a PhD in the Department of Biochemistry at Eötvös University. And her her main research topic is just drug discovery by pattern-based prediction methods, and she will be giving us a talk in this area.
Good afternoon. I will present you our novel in silico drug effect prediction method called drug profile matching, and I will try to reveal the contribution of 2D and 3D structural features of drug molecules in its prediction power. So first of all, what is the drug profile matching method? It exploits the information content of interaction profiles. Interaction profiles are generated by molecular docking of a drug molecule into a series of non-target protein binding sites. And for each drug protein complex, the lowest virtual binding free energy values are stored, and this is how we create the interaction profile. And it is important to note that we don't investigate the individual values, but this whole profile serves as a pattern that can describe the drug molecule. And we perform this interaction profile generation for all of the FDA-approved drug molecules, so more than 1,200 drugs. And this is how we got this matrix that we call interaction profile matrix. And every row in this matrix represents the interaction profile of a given drug molecule. And we also extracted medical effect information about the drug molecules, and we created the so-called effect profile matrix. In this matrix, a blue cell, this is a binary matrix, and one marks that we know that a given drug molecule has the investigated medical effect. And the white cells, so 0 means that we don't have information about that. So a little bit more detail about the effect database we used in our study. So this is an in-house hierarchical effect database that consists of 2 levels. There are main effects such as this one, antihypertensive agent. And it is divided into several subcategories, like you can see here, based on the mechanism of action. And in our database, we have 62 of such main effects, and they are further divided into the subcategories. And this way, we can investigate the impact of reducing wide effect categories into more cohesive subgroups. So let's get back to these 2 matrices. So we have the interaction profile matrix, which contains the interaction profiles of the drug molecules generated by molecular dockings. And we have the effect profile matrix, and we would like to identify the relationship between the interaction profile matrix and the given effect, which is represented as a column in the effect profile matrix. And we used multidimensional statistical techniques for this task. We applied linear discriminant analysis that can calculate probabilities for each drug effect pair. And this is how we created this new matrix, which is called effect probability matrix. You should know that in contrast to the effect profile matrix, the values in this matrix are continuous, ranging from 0 to 1. So basically, this is some kind of recalculation of the effect profile matrix, including the external knowledge of interaction profiles. And what we can do next is that we select an effect of interest, and we can sort the molecules by decreasing probability values. So for example, and that's how we get a so-called prediction list. This is an example of the ACE inhibitors, the subcategory of antihypertensive agent. And this is only the top of the list, so the molecules having the highest predicted probability values, but this list contains 1,200 molecules, so all of the FDA-approved molecules are ranked based on their probabilities. And I use here the same colors as in the effect profile matrix. So blue marks that this effect was already known for the molecule, and white means that it was not registered as an ACE inhibitor. And what we can do here is that we can define a cutoff value, like for example this one, and we can consider the molecules having greater probability than this actual cutoff value as positives, so having the investigated effect. And the positives can also divide it into true positives and false positives. And we can consider false positive molecules as predictions, because of course this result can originate from classification errors, but considering our limited amount of information about the complete medical effect profiles of drugs, they can also be actually true positives. And negative molecules can also be divided into two groups, the false negatives and true negatives. And based on this, we can create a measure of accuracy. We can draw a curve if we plot the true positive rate as a function of the false positive rate for each possible cutoff value. So here you can see the red line is for perfect classification because all of the positives appear at the top of the list and no false positives. So there exists a cutoff where all of the positives are recognized as true positives. And for the measure of accuracy, the area under this curve is used. So the best value is 1. And my other example, the gray line, is the so-called line of no discrimination, because here for every true positive also a false positive appears. And I'm showing some more ROC curves here, and these are real ROC curves we obtained by the drug profile matching method. And actually, the blue one is the worst one we got for our effects. So we can say that drug profile matching could excellently classify the drug molecules. But as you can see, this method does not use direct structural information of the molecules. And in order to understand how it works, we wanted to know to what extent can these results be explained by the 2D and 3D structural information of drug molecules. And this is the point where we use the ChemAxon tools for the two-dimensional representation of the molecules. We use the ChemAxon chemical fingerprints and calculated the Tanimoto similarity between the molecules, and we created a 2D similarity matrix. And for the three-dimensional description, we used the flexible 3D alignment tool of ChemAxon and calculated alignment scores. And these alignment scores were entered into the 3D similarity matrix. So then we wanted to calculate AUC values. So the similar accuracy measures we used for drug profile matching. And this was the screening protocol we applied. So we used these similarity matrices, and we had we used the same effect categories. And the screening protocol for a given effect looked like this. So first we selected an active molecule as a query structure, then sorted the database molecules by decreasing similarity, and that's how we got a list similar to the previous one I was showing to you, and calculated the AUC values to measure the accuracy. And because the conventional searches cannot handle multiple active query molecules, we repeated these three steps for each active molecule of a given effect. And then finally, we calculated the average AUC and the standard deviation. So this is how we created these measures, these accuracy measures. And here are the results for drug classification. So as I told you, drug profile matching could excellently classify the molecules, while the two-dimensional and there was no significant difference between the performance of the two-dimensional and three-dimensional methods. For all three methods, the division of the molecules into more cohesive subcategories could improve the classification accuracy as expected. At this point, I would like to mention that the aim of this study is not the direct comparison of the three methods, because drug profile matching is a machine learning approach. It is a more complex method, and two-dimensional and three-dimensional searches are more robust, and they have their place in the drug development process. At this point, we wanted to find out that for which effects does this docking-based method add extra predictive power. So for which effects is it worth using the more complex method, and which effects can be handled effectively by the traditional searches. So here are some examples. These are the rigid structural categories we examined here. The benzodiazepines and the glucocorticoids. And as you can see, there is no standard deviation for drug profile matching, because it is able to handle all of the active molecules in a single search. And so all of the three methods did very well for these effects, and almost perfect classification was obtained by all of the three methods. So we can conclude here that the more complex drug profile matching method is not required in these cases. Obviously, there is no need to dock these molecules to a lot of protein binding sites. But on the other hand, we also found that for structurally diverse effects, so these are all main effect categories, the ones I listed here: anti-inflammatory agent, anxiolytic agent, anti-glaucoma agent. The traditional two-dimensional and three-dimensional searches couldn't really handle these effects, but drug profile matching produced high accuracy values. And this is my last example. This is the main effect, anti-epileptic agent. Here you can see the subcategories belonging to this main effect. And you can see that this category is structurally very diverse, and drug profile matching was able to classify these molecules. So my conclusions are that the rigid structural categories can be handled effectively by the traditional searches, and the more complex drug profile matching method is not required in these cases. But we think that the main strength of drug profile matching is to screen wide effect categories containing structurally diverse molecules. And we have a possible explanation for this issue, but at this state, this is only a suggestion that because molecules are characterized via their interaction to non-target protein sites, we think that maybe biologically more relevant rare conformers can evolve in the binding pockets. And finally, here are the people involved in this project. In the middle, you can see my research group at the Department of Biochemistry, and we have two collaborators, the Semmelweis University and the PrintNet. And I want to thank ChemAxon for this opportunity to present my work, and thank you for your attention.
Hi, interesting talk. I did a similar analysis, and I wonder if you have considered a different performance measurement method, because the ROC IUC which you used has this problem that it doesn't consider early recognition, right? So there are a lot of cases where you can find a better ROC IUC or no significant difference among the whole data set, but you have a bad early recognition actually, because if you have a look at the curve, you see that it will descend after the, I don't know, 100 or 200 molecules, it will descend steeply, but you're missing all the important, for visual screening, important part of the list actually, because the first molecules are not active.
So actually, we know this drawback of ROC curves, and we also calculated the bedrock values. So and then we have found that that the drug profile matching can overcome this issue, because as you can see, the arrow seekers I showed you, so all of them were, yeah.
There was a funny thing in your slide.
Oh.
Did you notice that the name of a pharmaceutical or pharmacological effect was actually matching a structural class name? You called them benzodiazepines.
Yeah.
And all the benzodiazepines which were there, they were active, because otherwise the FDA would have said no. So take care of this huge bias. You know, there's zillions of other benzodiazepines which would be inactive, but they're not in your training set. So it's no wonder that the 2D pattern recognition tool can recognize the molecules that go by the biological effect benzodiazepines, because the only thing it has to do is to recognize a seven-membered ring.
Mm-hmm.
Now, on the other hand, all the things that end in a statin are active because they're FDA approved, but there are zillions of molecules that look like a statin and never bind to a kinase. So you cannot learn from FDA approved drugs only. You have to complete your set with reasonable decoys and run it all over again, and you'll see that a lot of your conclusions will change.
Yeah, we also did some external validation, but I didn't really have time to put it into the presentation. And we also did tenfold cross-validation on the results to prove the...
It's not a matter of cross-validation, you know. It's like the drunkard who says, man, I drank vodka soda, I drank gin soda, I drank whiskey soda and rum soda, and every time I was that drunk, now I'm going to stop drinking soda. It's not a matter of statistical tool, it's a matter of intrinsic bias in your data set. Those are all molecules that are approved. Okay. You need to address the entire chemical space without a bias, or else it will show up sooner or later.
Yeah, we are planning to extend. Thank you.